Okay, good morning, everyone. We're going to move from imaging to quantitative characterization of skin in terms of physiologically relevant parameters and using machine learning, obviously. So is this progression? No? How do I move forward? Okay, I'll just use this way. Okay, so the ultimate aim is, like I said, quantitative, non-invasive assessment of, say, relevant chromophore contents in human skin. And we achieve this by combining two optical techniques, namely diffuse optical spectroscopy and photothermal radiometry, and data analysis is based on inverse Monte Carlo approach. And we want to use this in the future for characterization of cutaneous lesions in terms of optimization of treatment, maybe for some research protocols, analysis of hemoglobin dynamics in traumatic bruises, and so on and so on. And the specific task I'm going to talk about this here today is how to bypass the computationally very intensive inverse Monte Carlo process with a predictive model employing a machine learning technology. So first I'm going to briefly describe the two experimental techniques used in this process, and then the data analysis. It relies on a four-layer model of human skin, optical model. And then I'm going to quickly describe the inverse Monte Carlo, how it works, and what are its limitations, and then come on to the predictive model, which uses the approach of random forests. And I'm going to show its testing of predictive performance using cross-validation and comparison with inverse Monte Carlo in a practical example. So these are the random forests. So DRS, everybody in this room knows. We are using this small integrating sphere for motion optics, and I just want to emphasize that for all those of you using these relatively small spheres, there's a considerable artifact involved called single beam substitution error, which arises due to the fact that between the time that you are calibrating your setup using a white reference and after replacing it with the skin, actually the light intensity and spectral content within the sphere actually changes. So you're not irradiating this sample with exactly the same light as you are irradiating your reference. And the instrument does not take this into account. And you have to explicitly remove this error. And this shows the difference between the uncorrected instrument-provided reflectance and the corrected one. So it's quite substantial, especially after you start doing inverse analysis by using uncorrected data. We've shown this earlier and how to remove it. In terms of data analysis, further on, diffusion approximation is known to fail close to the skin surface, so we're going to use Monte Carlo. This also allows us to remove, to account for the so-called red loss artifact, which means that due to the fact that this opening is finite, some of the longer wavelengths are actually escaping. They're not accounted for in your measurement, so you get this red loss, so-called. If you compare a one-dimensional solution, which corresponds to infinite opening, with the actual one, there's going to be some difference here. And by using Monte Carlo, we can certainly account for that as well. The second thing, though, is that in our previous assessment, we found that the data included in this spectrum, everything is lumped together, right? It's depth-wise. So as long as you have homogeneous material, that's fine. But if you have a multi-layered structure, everything is lumped together. So you really need to know how deep certain chromophores are in order to have a reliable quantitative assessment of their contributions. So to try to assess both from the same data set, it's just unreliable. It's not unique. So you need definitely some additional experimental approach. It can be spatially varied reflectance. It can be temporally resolved. You need something else, right? And in our case, this is this additional technique, which we like. It's called pulse photothermal radiometry. The basis is you use a millisecond light pulse, which is selectively absorbed inside your tissue, and then you collect the infrared emission of the surface after irradiation. And the point is, so we use a fast infrared camera, 1,000 frames per second, area of interest about 1 or 2 or 4 millimeters squared in the mid-infrared. And the point being that as you induce a non-uniform temperature profile inside your sample, inside your skin. For example, if you use green light, it's going to be epidermal melanin absorption and then the blood absorption deeper in. This is just a schematic. And then as this heat redistributes afterwards, according to the heat diffusion equation, the temperature close to the surface is going to undergo transient changes. and they reflect this depth distribution and the amount of absorption deeper in. The point is that, okay, sorry, now it works for some reason. The point being that it is possible, it's not so difficult if you know your initial temperature profile induced by the laser. It's relatively straightforward to compute. There's certainly enclosed formula to compute the signal, to predict the signal, but to solve the inverse problem, to go from the signal, the measured signal, to the reconstruction of the initial temperature profile is very ill-posed problem of solving this integral equation. It can be done, though, so this is one particular example of a lesion, hematoma, and the nearby healthy skin will give you very different these signals, radiometric signals. And then if we solve the inverse problem, we can get to the temperature profile induced by the laser in the healthy skin, indicating strong epidermal absorption and a weaker absorption in the underlying blood. And then in bruise, we see this enhanced absorption due to the larger accumulation of blood, obviously. Now, the thing is, this is already a non-unique and ill-posed inverse problem. And in some cases, it's very worthwhile to know exactly how deep the heat was distributed and how much the heating is. This is clinically very relevant, but however, if you want to now interpret these profiles in terms of blood content, oxygenation, melanin content, everything together, we should solve another inverse problem of how the light has or might have interacted with this structure, right? So, and this is our goal today. So how we go around this? We build this 4-layer schematic of skin, counting for the epidermis. Then we divide dermis into the superficial part, including the superficial vascular network, which we call papillary dermis. Okay, it's not maybe anatomically correct expression. And then the rest of the dermis, and then the adipose tissue underneath. And then we'll have these 3 parameters. So these 2 thicknesses, epidermal and endermal respectively, the melanin content, the 3 blood contents, and two different oxygenation levels. And then we also fit for the for the scattering properties of the skin, which also can vary on individual patient basis, right? So for the epidermis, we'll have the amplitude and the power, and for dermis, we'll have the amplitude and power. And also, it turned out in our earlier investigations, you need to count for the individual differences in the structure of the adipose tissue. It can have more fatty cells, it can have more connective tissue. It all reflects on the scattering properties, and this has to be adjusted in the individual as well. So taken together, we have 13 free parameters of our model. And this is our inverse Monte Carlo procedure. So by assuming some starting values for these parameters, we use MCML, Monte Carlo multi-layer model, to predict the temperature profile induced by the green laser pulse. From this, we then compute using that integral formulation the predicted radiometric transient, and then we repeat Monte Carlo 13 more times at specifically selected wavelengths to get a sense of this DRS. And then we compare this with measurement and close the loop, iterate until we are happy with the match. Right? So this is inverse Monte Carlo. And I'll just show you very briefly one example. We can get very nice fits. You see between the solid and dashed or dotted curve here, the experiment and fit respectively. And here are the assessed anatomical and physiological features. And they all fall within the expected regions or intervals according to literature. And here's a further demonstration of active manipulation using a pressure cuff. So this is the venous occlusion example. You can see the radiant contribution going up due to enhanced absorption, and the RS going down correspondingly. And here we have this pronounced increase in the blood content and reduction of oxygenation. And if you look at the other parameters, despite the fact that these 2 data sets were analyzed completely independently, it's quite robust in terms of epidermal thickness, dermal thickness, and so on. Everything is pretty much the same as expected. So this has just come up out in Biomedical Optics Express as of February this year. This is just a graphical representation of how this works. I'm going to skip this in the interest of time. So this is our inverse Monte Carlo. So we saw it works, but the challenge obviously is the time component. Okay, we made all the effort to massively parallelize it. So here you have the setup. So quite advanced graphics card, 2,500 GPU units working in parallel on Intel i7 personal computer. So for single wavelength, we get 2.7 seconds with 10 million photons. I think this is quite good, actually, right? Okay, so we are now here, a single Monte Carlo. Then we compute the signal. But altogether comes to 1 second to predict the radiometric signal, right? Then we have to repeat 14 more times to get a sense for DRS. So this is together, so this is about 10 seconds, 11 seconds, we get this pair. That's good enough, right? However, when you go into inverse analysis, first at each, so comparison is quick, right? It's no time, but when you want to improve, you need to know the gradients, so the differentials. So this means you have to compute them numerically again. So 14 more complete Monte Carlos to get the differentials in each step. And then you repeat this maybe 25 times, go around, you're at 1 hour, right? But now the result has a very high variance due to numerical noise, and there are the local minima that you want to avoid. So you repeat the whole process 5 times from randomly selected starting points, and you're at 5 hours. Okay, it's maybe tolerable for some things, for research, but certainly not very advantageous, right? So the idea here is now to have this predictive model which goes from provided experimental data pair to a predictor of result in one go. And I'll jump right to the bottom line. I'll tell you the results. We have done this, and the computation time is 0.3 seconds. And the variance in the results is considerably reduced as compared to Monte Carlo. So huge improvement in time. The technology is called random forests, as said. So the first decision was made to make separate random forests for each parameter. So we'll have 14 forests for 14 skin parameters. Each forest predicts one parameter. And then each forest consists of 100 decision trees, which obviously are single parametric. So the thing is that our data vector is provided to each tree separately, and this tree will be randomized somehow. And each of the trees is going to give one prediction of the parameter, and then they're all averaged out. So these decision trees are known to be very fast, but they tend to overfit. They have a large variance. However, by randomizing them and aggregating the results, you get a very quite accurate, I'll say, or result with a small variance and very little bias, which is the sort of out combination. So we are using 12,000 learning examples in this case. And so again, 14 model parameters, as I said, and each tree is provided with 175 features from the Monte Carlo simulated spectra. So the PPTR are compressed to 161 signal points, and these are the 14 DRS values at the strategically selected wavelengths, respectively. So the randomization is twofold. First is called bootstrap sampling of learning data, which means that every tree is grown from slightly different replica of the learning set. I'm not going to go into details. It's called bootstrap sampling with replacement, but each tree gets 12,000 learning examples, but they're slightly different between themselves. And then the second is more powerful. It's called feature subspace sampling. It means that at each node, at the time the trees are grown, each node seeps through all 12,000 data sets and tries to find out the optimal division point and division value, how this data set is separated into two subsets which have a lower variance. It maximizes this decrease of variance, but they are not all considering all 175 features. But at each node, we randomly select only 1/3 of these features, and then this decision point is made based on this information only. And then the next node is looking at another randomly selected 1/3 of the features. So some nodes are more sensitive to DRS, some more to PPTR, some more to the early times, to later times, and so on. This is how we diversify this forest. And aggregation just means averaging out this just polling across the whole forest, and this gives us the most likely predicted value, and as well as its estimate of the variance, which is also very nice. So cross-validation testing is the first thing one does. So here are results of 10-fold cross-validation, which means that each forest is grown anew from 90% of the available data, and the rest, the 10%, the holdout set, is used for validation. And you can then plot the actual value against the predicted value, and as long as they lie along the one line, you're fine. And then you can assess the variance, the effective error, okay, effective predicted error, just root mean square of the error in this direction, and here are the results. In absolute units, here are not normalized to the average value, and you see that some are on the order of 1%, which is really excellent. Some are up to 40%. But for the specialists in the field of machine learning, it's more relevant to consider not just this relative variance, but I mean the relative to a default model, which will always return just the average of the training set. So this is a very naive model. However, it already gives you something better than a random number. So the question is, how much better is our model against such a naive predictor, which will always return just the average, so this ratio. And this ratio being 1, it would mean we haven't learned anything other than just the average value, right? So it should be as low as possible. And these values, according to our colleagues, are quite good for this early stage development. And so this is our comparison now on a completely separate example. So this is on a person who was not involved at all in accumulating data for the training. So this is a completely different point in this multidimensional space. You can see the IMS is successful. And here's are the inverse Monte Carlo results and the predictive model results. You can get a sense that the numbers are very similar. And here's a graphical representation. So orange would be IMC result, including the confidence interval of estimate of at the 95% level. And this is the predictive model result. And you can see as we go from thicknesses, melanin contents, blood contents, scattering amplitude, scattering power, and blood oxygenation. Everything is very well within the error margins. So this is now an additional view in two-dimensional space. So say papillary blood versus reticular blood. Both models agree that papillary blood is higher than the reticular one, and oxygenation in the papillary is way lower, is around 40% versus the 80 to 90% in reticular. Scattering amplitudes in epidermis are much higher than the dermis. So this is now, again, the confidence interval of the predictive model, and this is the spread of the Monte Carlo data. So we would say that at this early stage of development, this is a very successful result. And remind you that the saving in time was a factor of about 60,000 in our case. However, Monte Carlo was run on a very capable PC with a dedicated graphics card, whereas the predictive model is run on a laptop computer at a single thread implementation currently. So effectively, we're talking 5 orders of magnitude speeding up of the result. So thank you very much. I'll conclude with this.